Take a look at this text. Individual letters, in isolation, carry no inherent meaning. Yet when arranged in a specific sequence and organized according to grammatical rules, they form structured and meaningful information. Rearrange them haphazardly or violate the syntactic structure, and the meaning either distorts or disappears entirely. A similar principle applies to consciousness and its disturbances. Neural coding is increasingly understood to rely not on single neurons, but on neuronal ensembles, transient networks of cells that fire together in a coordinated fashion at any given moment. Disruption of this integrative process can distort the brain's internal model of reality. Behavior based on a distorted model cannot be fully adaptive. Like any other physiological function, neural connectivity exists on a continuum and can deviate from the optimal level in either direction. In hypoconnectivity, the ability of neurons to form functional connections is reduced, resulting in an overly simplified and impoverished internal model of the world. In hyperconnectivity, excessive or aberrant linkages produce an overly elaborate internal model filled with spurious associations that have no counterpart in external reality. Because connectivity is critically influenced by neurotransmitter concentrations within the synaptic cleft, pharmacological interventions exert their effects in part at this level. When connectivity is insufficient, agents that facilitate synaptic transmission are employed. When it is excessive, drugs that dampen or block overactive pathways are used. This conceptual framework is particularly well illustrated by delirium. At first glance, the diverse medical conditions capable of precipitating delirium appear to share little in common. Acute myocardial infarction, sepsis, severe hypoglycemia, thiamine deficiency, anticholinergic toxicity, and many others. Yet each can converge on a common final mechanism, a disruption of effective neural connectivity. Consider acute myocardial infarction. A sudden drop in cardiac output leads to cerebral hypoperfusion and hypoxia. Sodium-potassium ATPase function becomes impaired, electrochemical gradients collapse, and neurons can no longer reliably generate action potentials. The result is widespread disruption of network function. Similarly, in sepsis, inflammatory mediators damage endothelial tight junctions, contributing to impaired microcirculation and cerebral edema, with consequent disruption of neuronal signaling. The function of the sodium-potassium pump depends not only on the presence of oxygen, but also on glucose and essential cofactors such as thiamine. Thus, metabolic insults, whether hypoglycemic or related to cofactor deficiency, can impair neuronal firing. Connectivity, however, is not limited to action potential propagation along the axon. It also depends on successful synaptic transmission. Any substance that impairs neurotransmitter release or postsynaptic receptor function can provoke delirium. In this sense, delirium can be viewed as a final common pathway for a wide array of systemic insults. At the cellular and network level, the core pathophysiology of delirium involves either impaired generation of action potentials or disruption of synaptic transmission. Both ultimately lead to disintegration and disorganization of functional neural networks that support coherent consciousness.